University of Nairobi. Dr. Marianne Mureithi, who is one of the researchers and scientists at COV. I'm also at the Department of Medical Microbiology. And in the panel, we have Professor Omu Anzala, who is a senior scientist at COV and former director. We also have Dr. Daniel Muema and Dr. Robert Langat, who are postdoctoral fellows, senior postdoctoral fellows at COV Institute of Clinical Research, who will take us through the immunology of SARS-CoV-2 and the different tests, the serological assays, antigens, and antibodies that are currently available. And we look forward to a great discussion and interactions with all of you. So without further ado, I'd like to welcome Professor Omu Anzala.
Thank you so much, Marianne, for the introduction. I would like to thank KNH team for the continuous ability to keep all of us informed. And I won't say much other than to say that when we hear about COV, at times my name has been there a lot, but I'm actually never alone. There's a huge team behind me, as you can see. And I'm happy to say that this is the team that I work with. This is the team that I've actually been mentoring. So I'm happy to see that I'm beginning to hand over the baton to the next generation. So without too much discussion, let me hand it over back to Marianne to lead the discussion, and let's all join in. Let's ask very, very hard questions, the kind of questions we cannot answer, because that's what pushes science to the next level. Over to you, Marianne.
Thank you, Professor Omu Anzala. Again, that's a good way to kick off this session. Yes, let's ask very hard questions, but more so let us learn and push the science way ahead. So without further ado, I want to welcome Dr. Daniel Muema. Dr. Daniel Muema is a senior postdoc fellow at COV Institute of Clinical Research, who's done several years of research of HIV and other viruses. He's a virologist in our team, who's also done some training in South Africa with a team from the University of Harvard University, at the Reagan Institute, at MIT as well in the US, and is now back in Kenya to take up this position at COV, and he'll take us through the immunology of SARS-CoV-2. And after that, we followed by a session of few questions. And then we welcome Dr. Robert Langat, who is also a postdoctoral fellow at COV. So Daniel, the floor is yours. Welcome. I probably want to ask the host to please make Daniel also a host so that he can share his slides.
I can see Daniel is already there.
Yes.
Yeah.
Daniel, you can share your slides. Daniel, if you're speaking, you may be on mute.
Maybe we try to reach him over the phone, Marianne.
Yes, so please, KNH team, please make Daniel the host. He's unable to share his screen. Daniel, you can hear us?
Marianne, can he communicate using the chat?
No, he's telling me he's unable to do that. You can only see raise hand. So he needs to be made a host.
I can see his own chat. He's saying I can hear.
Yeah, he's saying he can hear and he's there. So we can
He's the co-host now. He should be able to.
Oh, great. Thank you.
Daniel, you're now co-host. You can share your slides and please unmute.
Trying to reach is currently.
Hello, now this looks like it. Sorry, I just had to log out and come in again. I hope you can hear me now. Hello?
Yes, we can, Daniel.
Yes, we can hear you.
Sorry about that. I don't even know what was happening because I just needed to log out and come back in.
Okay, Daniel, the floor is yours. Please take us through this first session of the immunology of SARS-CoV-2. Welcome.
Okay, thank you. Thank you very much. Sorry for that, that technical itch. Yeah, so I'm going to discuss very basic concepts about the immunology of SARS-CoV-2 and how some aspects of the immunology of SARS-CoV-2 are being harnessed to manage COVID-19. So just as a quick reminder, just a very brief reminder on the composition of the immune system, the human immune system. And in the immune system, we have what we call the innate immune system, the innate arm of the immune system, and we also have the adaptive arm of the immune system. And in the innate arm, we have some cells that might be familiar to you, like basophils, neutrophils, eosinophils, natural killer cells, and also macrophages and dendritic cells, while in the adaptive arm, we have B cells and T cells mainly. And we have some crosstalk between the two arms because the innate arm tends to play a major role in the activation of the adaptive arm. And therefore, the quality of innate immune responses usually affects the quality of adaptive immune responses. So the innate arm is called innate because those cells don't require any maturation, any major maturations so as to control the invading pathogen. And they're always ready to help in controlling the pathogen of interest. While in the adaptive arm, the B cells and T cells usually need to undergo some level of maturation so as to fight the invading pathogen more efficiently. And the other thing that I would like also to mention before we go into the other slides is that there's a time component as to the responses that happen when you compare the innate immunity and the adaptive arm. The innate arm, because it's always ready, it tends to happen earlier, and the adaptive immunity arm tends to kick in much later. So the innate arm of the immune system tends to lead to production of a lot of cytokines, and cytokines are molecules that in one way or the other modulate the immune response by coordinating the immune response, coordinating the responsiveness of various immune cells. And so one of the major characteristics of COVID-19 is what we call a cytokine storm. And this one is characterized by a surge of many cytokines in a dysregulated manner. And in as much as these cytokines are usually useful during an immune response, if that secretion of cytokines, if that production of cytokines is dysregulated, it could lead to most of the pathologies that have been seen in COVID-19, and therefore the cytokine storm has actually been suspected to be a major contributor of pathology in COVID-19, affecting all the organs that have been shown to be affected in COVID-19 due to that unregulated excessive immune response, which is mainly thought to be mediated by the cytokine storm. And of course, if the cytokine storm is thought to mediate some of the pathologies, there are attempts to control that cytokine storm in COVID-19, and that has actually proven to be beneficial to COVID-19 patients. And there are some immunomodulators that have been used for this purpose, like dexamethasone, which has been shown, which is a very well-known immune suppressive drug, which suppresses several pathways in the immune system and could dampen the cytokine storm, which contributes to the inflammatory response in COVID-19. And it has actually been shown in some randomized clinical trial that people who receive dexamethasone are less likely to die when compared to people who receive the usual care, suggesting that modulating this excessive inflammation could be beneficial in COVID-19. And there are also some other targeted strategies, like targeting various pathways of the innate arm of the immune system. And one of them entails targeting the IL-6 pathway, because IL-6 is one of the cytokines that has been shown to be elevated in the cytokine storm that is induced in COVID-19. And therefore, there are some drugs like tocilizumab, which are meant to inhibit that pathway by blocking the IL-6 receptors. And some studies have actually shown that tocilizumab and serolumab, which are IL-6 receptor antagonists, actually reduce mortality by enhancing survival of patients who receive it when compared to people who don't receive it. So, but now that was about the innate arm of the immune system and how COVID-19 falls within that, how the immune responses to COVID-19 take place within the innate arm. But now, looking at the adaptive immune response to COVID-19, and the one that people talk most about are the antibodies, the B cell arm, and B cells, as you might know, are the ones that produce the antibodies against COVID-19. And the reason why we think antibodies are important in COVID-19 is because we think antibodies can mediate neutralization of the virus. And that's the most interesting, that's the most studied function of antibodies in most of the viral infections. And in fact, even most vaccines work by inducing neutralizing antibodies. So neutralizing is whereby if you have a virus, the antibodies are able to come and bind onto the virus and prevent that virus from accessing the target cells by preventing the virus from interacting with the receptors in the target cells. Also, neutralizing antibodies can also allow the virus to bind to the target cell, but bind in a manner that in one way or the other still inhibits the ability of the virus to infect the target cells. But there are other functions of antibodies that are of interest to us even in COVID-19. And those functions of antibodies are mediated by the other end of the antibody that does not bind the invading pathogen. And that end, we call it the FC receptor portion, the FC portion. And then there are various immune cells that will have receptors for that other end of the antibody. And therefore, the antibody can act as an adapter between the virus and the other immune cells, and therefore enable those other immune cells to clear the virus, like, for instance, by phagocytosing the virus, or even phagocytosing cells that are infected with the virus. And also by marshalling other cells that are able to kill infected cells, for instance, natural killer cells. So the antibodies could act as an adapter between an infected cell, which is expressing viral antigens on its surface, viral proteins on its surface, and a natural killer cell, and therefore enable the natural killer cell to come and kill that infected cell, and therefore stop the replication of that virus. So we think those are various ways in which antibodies could be beneficial in COVID-19. And natural COVID-19 infection has been shown to actually induce production of antibodies in a pretty fast way, in the classical way in which antibodies would be elicited. This is showing how binding antibodies were detected in a particular cohort when compared with the samples collected before the COVID-19 pandemic. And this was in people who are confirmed to be PCR positive. And most of the people by 20 days had experienced the maximum level of antibodies that they could produce. And then the antibodies started decaying over time because these people had now recovered. And then there is now what we call the neutralizing antibodies. And these are antibodies that we have confirmed in functional assays that they are capable of neutralizing the virus. And you can see that the kinetics are similar to the binding antibodies. And there's usually very close correlation between the neutralizing antibodies and the binding antibodies that target the spike protein that attaches on target cells. But something that has proven to be quite interesting is that in COVID-19, the people who have severe disease seem to have more antibodies. This cartoon is just summarizing the development of neutralizing antibodies over time in people who have COVID-19 after those people are stratified based on their severity of disease, with green showing the mild disease and red showing the red showing the severe disease. And if you look at these lines here, you can see that the red line for severe disease shows that those people elicited antibodies earlier and probably even higher levels of antibodies compared to the mild disease, suggesting that antibody response is probably dependent on the amount of antigen, which is dependent on the amount of the magnitude of the infection that someone is carrying. But antibodies are still thought to be very effective in as much as they are higher in people who have more severe disease. They are still thought to be effective. And in fact, that has been utilized, where some monoclonal antibodies have been isolated from COVID-19 patients. And those monoclonal antibodies, like these two, otezumab and bamlanivimab, have been isolated from COVID-19 infected patients, and they have been expressed as monoclonal antibodies, recombinant monoclonal antibodies, and used to control viremia in people who are already infected with COVID-19. And therefore, as you can see in the blue line, where some people got a combination of these two SARS-CoV-2 monoclonal antibodies, they had lower viremia compared to the placebo group, which is shown in red, which did not receive these monoclonal antibodies. And then the other groups lied in between. Of note, the cycle threshold, the higher the cycle threshold, the lower the viremia. So the blue line, which is for the people who received the antibodies, had lower viremia. And then there's the other arm of the adaptive immune system, which I mentioned earlier, which is the T cells, which together with B cells comprise the adaptive arm of the immune system. And T cells are divided into CD4 T cells and CD8 T cells. And CD4 T cells are the ones that coordinate various immune cells and provide help to B cells and also to CD8 T cells. And they do this after receiving antigen presentation from antigen presenting cells like dendritic cells and macrophages. So you can see that CD4 T cells lie at the center of the immune response, and they are therefore capable of impacting all other components of the immune response against COVID-19. While on the other hand, CD8 T cells are cells that are cytotoxic in nature, and they are able to go and identify infected cells. So these would be cells that are infected, like epithelial cells that are producing SARS-CoV-2. And then the cytotoxic CD8 T cells would come and kill those infected cells. And once those cells are killed, it means that you have controlled viral replication, because you have curtailed viral replication in those infected cells. So we think that these cells actually play a role in SARS-CoV-2 infections. And in fact, a few studies have managed to look at the development of T cell responses over time during the early phases of infection. So in this case, in this particular study, they were able to stratify the patients based on whether they are mildly infected or whether they were severe cases. And they were able to show that generally T cells, they were induced very early during the infection. And something that was quite interesting was that the mild patients had higher levels of functional T cells compared to the severe cases. Some are suggesting that these T cells could be able to control viral replication. And maybe that's the reason why people who had higher T cell responses ended up having milder disease compared to people who had lower T cell responses. But this is still something that is being investigated. So just as a summary of that, In that study, they were able to see differences between severe cases and mild cases of COVID-19, where the mild cases had more functional T-cell responses compared to the severe cases who had lower levels of functional T-cell responses. So maybe then we can then ask, can these naturally acquired immune responses prevent someone from reinfection with SARS-CoV-2? I think that's one of the biggest questions everyone is asking in their minds right now. Now that we know that these immune responses can be elicited in natural infection, can they actually protect us from future infections? And the reason why we ask that question is because we know that the adaptive arm of the immune system, which comprises of B cells, which produce antibodies, and also comprises of T cells, that adaptive arm of the immune system is capable of creating memory. And basically what we refer to as memory is can be described as I'm going to do right now. When you get your primary exposure, let's say you get your first infection with SARS-CoV-2, you'll generate, after a few days, you'll generate T cells and B cells. And in this case, I'm showing antibody responses, which are B cell responses, basically. After some time, you'll generate those antibody responses. And then once the infection is cleared, the antibody responses will decay, but they'll not decay to below detection. They'll remain still detectable, and you'll keep some antibodies still circulating. You'll also keep some memory B cells still circulating. And these cells are there just in case you get a secondary exposure, you get another exposure to the same pathogen, in this case, SARS-CoV-2. It means that you'll not be starting from zero. And that's the reason why in the event that someone gets re-exposed again, those memory B cells and memory T cells are expected to mount a faster and a stronger immune response. And that's the reason why subsequent reinfections might be mild or might be prevented altogether, especially because you also have antibodies that are circulating and that could neutralize any invading SARS-CoV-2 pathogens. And so based on this, you would expect if someone has been infected with SARS-CoV-2 previously, you would expect them to be having some immunity against any future re-exposures, either sterile immunity where they don't get infected at all, or even if they get infected, and maybe future infections could be mild. And this was one of the big questions that was being answered at the beginning of the pandemic. And what these guys did, this group, what they did is they decided to look at that, whether immune memory can protect against reinfection, but here they used non-human primates, monkeys. And what they did is they took some convalescent animals, then they took the antibodies from those animals and they transferred them to a new set of animals that had never been infected with SARS-CoV-2. So animals that had been infected with SARS-CoV-2 and recovered were taken and infused into animals that had never seen SARS-CoV-2. And then, so that was this group, the one that received antibodies from the previously infected group. And then there was a group that didn't receive antibodies. And so they then challenged these two sets of animals with SARS-CoV-2. And what they saw is that the animals that received antibodies from previously infected animals, that group was protected. If you look at this, this is a viral load, and that group was protected, unlike the group that had not received antibodies from previously infected animals. So suggesting that antibodies that are elicited during natural infection could protect from future infections. And then they also wanted to look at whether CD8 T cells could also protect from future infections. And what they did is they allowed some animals to stay for a while until the antibodies had decayed. And then in those same animals that had been previously infected, they depleted CD8 T cells using some other CD8 depleting agents. And then, so there are some that were not depleted for CD8s and others got some depletions of CD8. So they basically, their previous induced CD8 T cell responses were wiped out. And when they were rechallenged with the SARS-CoV-2, the ones whose CD8 T cells had been wiped out, some of them got some transient infections, suggesting that in the absence of adequate levels of antibodies, CD8s could also protect. So suggesting that immune responses that are elicited during a natural infection could protect from future infections. And so when we come into humans, there's a very nice study that was done in UK, where they showed that if you take people who had been infected during the first wave in UK, those are the people who are shown in blue here, and then you follow them into the second wave, and then you compare them with people who had not been infected during the first wave. Most of the infections that were happening in the second wave were happening in people who had never been infected with SARS-CoV-2, suggesting that these people who had been infected with SARS-CoV-2 in the first wave were somehow protected in the second wave, even though some of them still got reinfections, but they were much lower than the ones who had never seen the virus before. So knowing that immune responses that are elicited in natural infections can protect from future infections, the other question that would come up is, what about vaccines? Because with vaccines, we try to recapitulate the immune responses that are elicited in natural infections to see whether those vaccine-induced immune responses could protect from infection. And so just a quick mention on the various vaccine platforms that are being used for COVID-19. We have what we call the inactivated vaccines, whereby they take the SARS-CoV-2 virus itself and inactivate it chemically to ensure that it cannot cause disease, it cannot infect the person, but it is able to elicit the immune responses that are required. Then we have the traditional strategy of designing vaccines whereby recombinant proteins are used. And in this case, people would take the spike protein, which is the one that the virus uses to attach onto target cells, and then express it as a recombinant protein, or even on top of that, even formulate it into a virus-like particle, which only has the recombinant proteins on the surface, and therefore use it to try and elicit an anti-COVID immune response in a healthy person. And then there's also what we call the viral vector vaccines. And this include like the vaccine which is used by the Oxford AstraZeneca group and also the Johnson & Johnson group. And this is where a virus, and a virus, in most cases, a virus which has been modified so that it is non-replicating, like the adenovirus, some chimpanzee adenoviruses, or even some human adenoviruses, could have an insertion of the spike gene. The spike being the protein that the virus the SARS-CoV-2 virus uses to attach to target cells to infect them, and therefore it means that this virus, this vector, can carry that gene into the cells of the vaccine recipient, and therefore that gene can then mediate the production of the actual protein in the body, and therefore lead to an elicitation of an immune response. And then you have the other platform, that is the mRNA vaccines, where you actually package that spike gene in lipid nanoparticles and deliver them into the body so that that spike gene can also go and do the same to lead to production of the spike protein, and after that elicit an immune response. And also the DNA vaccines, where you have circular DNA that is also encoding the spike protein to go into the cells and also be able to induce an immune response. So I just thought of showing this slide again because of just to remind ourselves how it would be in a vaccine setting. So in a vaccine setting, you are basically trying to recapitulate what happens in a natural infection, where you give someone a vaccine, and then after a few days, that person will mount what we call a primary immune response. And then after that, some of it will decay, but they'll maintain immune memory, both in form of antibodies and memory B cells and also memory T cells. And then you expect that if that person ever gets a natural exposure to SARS-CoV-2, they'll be able to mount a very strong immune response very fast, and that will be able to clear the infection or even prevent infection altogether, especially because you'll be having antibodies that are remaining in circulation during this time before the person is exposed. And so some of the vaccines are already in clinical use, at least have received emergency use authorization, like the Oxford AstraZeneca vaccine, the ChAdOx1 nCoV-19 vaccine, which is in use here in the country. And these vaccines have been able to elicit the expected immune responses. So they are immunogenic in that they are able to elicit the desired immune responses. So this is showing what the ChAdOx vaccine is able to induce. If you have people who are vaccinated with either 2 doses or 1 dose, you can see that the 1 dose group shown in dotted lines, they are able to induce immune responses, but not to the level that could be induced by the 2 dose regimen, which is shown by the 2 lines, and the different colors are showing different age groups, suggesting that this vaccine is immunogenic across different age groups, and there are no differences between those age groups. So because this vaccine is able to induce good levels of neutralizing antibodies, of course, you would expect it to be able to protect from infection, and indeed that's the case. So on the right, I'm showing the efficacy of the same vaccine, and this was in one of the clinical trials across different countries. This is the aggregate data, and the blue is showing the placebo group, which received a meningococcal vaccine, while the red line is showing the group that received the COVID-19 vaccine. And when you look at the cumulative proportion of participants that got COVID-19, at least even mild COVID-19, you can see that the group that received the vaccine was generally protected from COVID-19 compared to the placebo that received the other vaccine, which is not a COVID-19 vaccine. And the same to the mRNA vaccines. And these vaccines are also quite immunogenic. And here I'm showing the one from Moderna. And there's also the other one from Pfizer, which has similar immune responses and similar efficacy. So this Moderna vaccine was able to induce neutralizing antibodies. After the first dose, there was an initial induction of antibodies. And then after the second dose, there was also even a greater boosting of the immune response. And you can see what happens during second exposures to the antigens. And so this vaccine was capable of inducing very high levels of neutralizing antibodies. And as you would expect, when it's able to induce those levels of antibodies, it was protective. When you compare the group that didn't receive the vaccine versus the group that received the vaccine, there were more COVID-19 cases in the group that did not receive the vaccine compared to the vaccine group. And I've showed you the immunogenicity of the vaccines with regard to induction of neutralizing antibodies. But something that we need also to remember is that these vaccines are able to induce T-cell responses. So this one is showing the ChAdOx nCoV-19 vaccine, the AstraZeneca vaccine. And it's showing that when you do ELISpots to try and detect T-cell responses, functional T-cell responses, you can see that after the first dose, there was induction of T-cells, and also after the second dose, those T-cells were still there. And remember earlier on, I showed you that T-cells usually function by killing infected cells. And so this means that even in breakthrough infections, these T-cells that are induced by these vaccines might be able to kill infected cells and therefore modulate the level of viral replication. And that might give some benefits by modulating the severity of disease, even in breakthrough infections. But then we have the other problem that has been quite widely debated on, which is the issue of reinfections. And with regard to reinfections, I'm going to show you data on 2 possible causes of reinfections. The first one being the decay of immune responses, and the second one being the emergence of immune escape variants. And so when you think about a classical immune response, I showed you this earlier, where I showed you that antibody responses develop quite well in people who have COVID-19, natural COVID-19 infection. And I also showed you antibody responses after vaccination. But when you look at antibody responses after natural infection, once someone recovers from infection, those antibody responses start decaying because there is no antigen to continue driving the production of those antibodies. And so you would expect after a prolonged period of time, those antibodies will decay to levels that are below detection, and probably levels that are below protection if those antibodies were actually the ones that are conferring protection earlier on. So this is something that is expected to happen. And then there's the other issue of emergence of immune escape variants. And the best way to show this is with the data from South Africa, where the emergence of new variants has been very well characterized. So on the far left, if you look at the viruses that are circulating in the second wave compared to the viruses that are circulating during the first wave, it has been shown that actually in the second wave, there was emergence of some mutations, like the 541Y.V2, which is also referred to the B1351 variant. So that variant emerged in the second wave, and the emergence of these variants, of course, is usually expected to be driven by some pressure, and some of that pressure could be pre-existing immune responses. So what this group in South Africa did, they then took the virus that, the second virus, the emerging variant, and also the ancestral virus that was circulating in South Africa. And then they also took antibodies, or rather plasma samples, which contained antibodies that were elicited during the second wave, and also antibodies that were elicited during the first wave. And so they wanted to see whether antibodies elicited during the first wave and the second wave, they were able to neutralize each of those two viruses. Especially because they were concerned about the second variant, which had emerged in the second wave, whether it would be neutralized by the earlier antibody responses. And this is what they were able to show. If you take viruses from the first wave and you try to neutralize them using samples, plasma samples, antibodies that were collected in both the first wave in orange and the second wave in blue, that virus was susceptible to both those two sets of samples. But if now you took the second virus, the second variant that came much later, and you try to neutralize it with antibodies from the two waves, that second variant was only neutralizable by antibodies that were elicited in the second wave. And it was resistant to antibodies that were elicited in the first wave, suggesting that the variants had escaped from immune responses that had been elicited, that had been induced in the community during the earlier phases of the pandemic. And so it also means that the antibodies that are produced in the second wave are broader than the antibodies that are produced in the first wave. And if the early ancestral virus ever comes back, it will find people who already have antibodies that can neutralize it in as much as they were induced much later on. So these two concepts, both the decay of immune responses and the emergence of immune variants, could also modulate protection that is elicited by vaccines, and therefore could also lead to reinfections or later infections in people who have been vaccinated. Then the other question that I've had people ask around is, if I have had COVID in the recent past, should I still go and get vaccinated? And I thought we can show you the data so that the data can talk for itself. And so here, there's a group in the US that took two sets of people who are receiving the Pfizer vaccine. And one set people who had never been infected with with SARS-CoV-2, they have never had COVID-19. And another group had COVID-19 in the recent past and they had recovered. So the group that had previous COVID infections, or of course had antibody responses against SARS-CoV-2, and the group that is shown here in red, at the time when they were receiving the vaccine, they already had immune responses. And then the other group, of course, didn't have immune responses. And when these two groups were given the vaccine after the first dose, the group that had previous COVID-19 infection had very good antibody responses after just one dose of the vaccine. And getting a second dose of the vaccine did not seem to give extra benefit, but the first dose seemed to do the magic. But now the other group that had never had COVID-19 previously, they mounted a modest amount of immune response after the first dose, and then after the second dose, that's when they were able to catch up with the other group that that had had previous COVID. This on the right is just a summary of that. So suggesting that if someone has had COVID previously, they'll benefit a lot from receiving the vaccine. But the question is, how many doses of the vaccine do they need to be able to get maximum benefit? But it looks like they would still benefit, and that's the reason why this data suggests that if you have had COVID before, it's still very important to get the vaccine so as to get the highest antibody titers that are similar to someone who has gotten 2 doses of the vaccine and they have never had COVID, and then get maximum protection. So the other thing that I thought would be of interest is the adverse events of interest. And in this case, I'm going to talk about thrombotic thrombocytopenia, which has only been recently described in people who have received some of the COVID-19 vaccines. And this is a very evolving debate, and the data is very scanty. There are some proposed mechanisms. And the particular mechanism that I'm showing here is something that is expected to evolve after more data is available. This mechanism is informed by the fact that those people who have shown thrombotic events after receiving COVID-19 vaccines, they seem to have high levels of antibodies against what we call platelet factor 4. And this platelet factor 4 is produced by platelets, and it is it plays a role in another condition called heparin-induced thrombocytopenia, and in that case, it usually works hand in hand with heparin to induce thrombosis. So this was a modification of that model for heparin-induced thrombocytopenia. And so if this platelet factor 4 is produced by is released from platelets, it would bind to the antibodies that have been shown in these people who are reacting to the vaccine and form immune complexes that can then go and bind to the receptors, other receptors in the platelets, which we call the FC receptors, which recognize the other end of the antibody, and then activate the platelets. And that could lead to thrombocytopenia. It could lead to activation of those platelets and leading to thrombosis. So this is a very early version of what we think, and it could change anytime with the new data, but I thought it would be a good thing to discuss today. So with that, I would like to acknowledge various people whom we have discussed this presentation with, and also COV and the University of Nairobi. Thank you.
Thank you very much, Dr. Daniel Mrna, for a very clear presentation, especially on the immunology of the SARS-CoV-2, the immunology of the vaccines, the vaccine immune induced responses, what it all means, and also even touching on the adverse events of interest. So that is clearly explained, and we want to thank you. So we would allow a few questions, and because of time, we'll not allow many. There are several on the chat, and there are a few raised hands. So allow the people, the 2-3 raised hands, we'll give you an opportunity to just quickly ask your question, and we'll quickly go to Dr. Bosire. You can quickly ask your question.
You've unmuted.
Yes, Dr. Bosire.
Sorry, I was driving. It was by mistake. Thanks. Thanks.
Okay.
Yes.
I actually see some questions in the chat.
Yes, so let's go to the chat as the host unmutes some of the hands that are raised. So we could quickly go to an interesting question from Peter. How does pre-existing HIV/AIDS infection interact with the SARS-CoV-2 infection immunologically? Can you answer that?
Yeah, this has been a question of interest, especially in Africa, where we have quite a significant proportion compared to other places in the world where we have a significant proportion of HIV-infected people. And I have to say that the evidence is still a bit shaky, but there are some studies that have shown that people who have HIV infection are more likely to die from SARS-CoV-2 infections. And that would imply that the immune suppression that is induced by HIV might impair immune responses against SARS-CoV-2. There is also a study that is not published, but I know has been done in South Africa, where if you look at people who are on ARVs and you compare the antibody responses between them and people who are not HIV infected, you don't see much of a difference. You only see minor trends, but you don't see much of a difference. And there's also another study in South Africa which earlier on showed that people who had HIV are more likely to die from COVID-19. So I would say that it's likely that HIV impairs COVID-19 immune responses, and therefore, and that is probably more significant in people who have not controlled their HIV viremia.
Okay, thank you. There are several questions on how to improve the immune responses without vaccinations. There's one asking about T cells to boost their functionality. There's also a question on the role of Tregs. I know we had discussed that. Tregs in managing the cytokine storm. So Dr. Mama, maybe you can allude to that a bit.
Yeah, the ability to improve T cell function without vaccination. That's a very interesting question. Like in HIV, of course, the first thing you'd say is if someone has suppression of HIV viremia, because we know HIV impairs T cell responses. So that could be a way of improving T cell responses without vaccination. But you'll not be improving SARS-CoV-2 specific T cell responses before you get infection with SARS-CoV-2, or even before you get vaccinated. So that will just be a general strategy to improve T cell responses generally. There are many studies about how you can modulate the immune system by nutrition, but those studies it's a very new field, and I don't think anything tangible has yet been concluded from those kinds of studies. So I wouldn't go with any current evidence on that. We would need to wait. What was the second question? Sorry.
So there are several questions actually now on antibodies, how protective after the infection, and if in cases of those who got infected after vaccination with the AstraZeneca vaccine, so what was the ability of those antibodies? Were they protective enough? I know you've shown some immunology of the variants and how vaccine immune escape occurs, but maybe you can just discuss this a little bit more, the functionality of these antibodies, especially after vaccination, and how long is the protection induced will last?
So we know the antibody-induced responses can last several months. Of course, we have not had enough time to look at how long that can last, because the vaccines are just are only recently developed. But some studies have shown that even after 6 months, you will get adequate levels of antibodies in people who have been vaccinated. In people who have had natural infection, it is very variant. The variation is very huge, because people who have had only mild infections, they seem to develop very low levels of antibodies, and therefore those antibodies, they tend to disappear even within 3 months or so below detection. But of course, that does not rule out whether there are some other components of the immune system that could still confer protection to those people. But as in the animal model study that I showed you suggested, is that antibody responses could be protective for quite some time, but at some point, other components of the immune system would have to take over, if at all they are there in that person, to confer protection. And then with regard to the emerging variants, there's a study that was done in South Africa to look at whether those antibodies were able to neutralize, the one that I was showing you, whether those antibodies were able to neutralize the new variant, and the antibodies and that was even after vaccination. The one I showed you was for naturally elicited antibodies, but even after vaccination, the antibodies had lower ability to neutralize the new variant. However, and that was associated with the diminished effectiveness of the vaccine. However, in that clinical trial, they didn't look at severe disease, whether the antibodies that were induced by the vaccine, in as much as the virus had escaped, were those antibodies still able to prevent severe disease? Or were the T cells that could have been induced at the same time as those antibodies, when those antibodies were being induced, could those T cells reduce the severity of infections with the new variant? So that was not clear. And therefore, they only showed that the diminishing effect of those antibodies against the new variant was associated with infection in people who had received the vaccine, but they didn't have enough power to look at severe disease. And then I saw, there is a question that I answered in the presentation, how many doses people need if they have had previous COVID. So we, for logistical reasons, we are not able to identify everyone who has had previous COVID infections, especially the mild ones that went unnoticed. And so that's the reason why everyone is still getting 2 doses for logistical reasons, and it will probably be cheaper even that way. But that's a debate that is only starting to be addressed now, and these data that are coming out could inform policy on whether people need to be tested before receiving a COVID-19 vaccine, and so that information can inform on how many doses they can receive.
Thank you very much. The very last question as you take down your presentation and the next speaker comes up, there are several questions asking then, when should you take the vaccine, especially after following recovery of infection of the COVID-19 disease? So do you have a recommendation? I know you mentioned it and you showed some evidence, but maybe you can talk more about that.
Yeah, so during someone who has only had COVID-19 infection in the recent few days, in the past few days, would be expected to have a very dysregulated immune system. As I've shown you this, what we call the cytokine storm, and so many things are happening to the body. So that person's immune system might not be ready to maximize on the benefits of a COVID-19 vaccine. So what has been suggested is for people to wait after you recover from COVID-19, to wait for at least 4 weeks or so before you can go and get a COVID-19 vaccine. And also, it has also been shown in some of the things that are being tried in clinical trials that if you leave a gap, the longer the gap between the first exposure to the antigen and another exposure to the same antigen, the longer that gap, the higher the immune response that is elicited in the second exposure. And that would be the case also in a situation where you have viral infection and then subsequent vaccination. So leaving that gap a little bit longer might enable that person to maximize the benefits of being vaccinated.
Okay, thank you very much. And there's one
Marianne.
more. Yes.
Marianne. Hello.
Yes, yes, Prof.
This is Ndavi Muia. Hello.
Yes. Good evening, Professor Ndavi.
A quick one to Moema. First, thank you, Moema, for that presentation. It's very, very enlightening. My question, post the infection and the administration of the vaccine in the studies that you presented, did we look at the duration between when the infection occurred, when someone, in quote, recovered, and when now that first dose was given? Did we control for that? So that then we look at the evolution of the immune response, as you showed. Did we take care of that duration in the studies? Thank you.
Thank you. That's a very good question. And the thing is, yes, that was not controlled because these people had had previous COVID-19 infections at varying times prior to their vaccination. So this was a study that was just piggybacked on the vaccination activities that are taking part in various parts of the world. So it was an opportunistic approach of answering that kind of question. And maybe some studies that look at that in a more systematic way will be coming up soon.
Thank you.
There's so many other questions, but because of time, we'll allow the second speaker and some questions on the assays that are rolled out, that are being used, and how adequate they are in measuring responses. So allow me to welcome Dr. Robert Langat, who is a postdoctoral scientist at KOV Institute of Clinical Research, which is at the College of Health Sciences. Robert has been a virologist, immunologist at the Institute, and has done his research in collaboration with the Northwestern University in the US, Imperial College London, and he's here to talk about the assays, antigens and antibodies. And well, Robert, we want to welcome you. The floor is yours. Any other questions, please put them in the chat. We'll be able to go through them at the end of this session. Robert, welcome.
Thanks, Marianne, and thanks, Dr. Muli, for that enlightening introduction. So the talk of Mrna laid the foundation for my talk. So here we are looking at the various assays that you can use to diagnose or detect the SARS-CoV. So a brief of the virus itself. So this virus is an enveloped virus, which is a single-stranded RNA genome. It belongs to the 4 coronavirus genera. So we have 2 in mammals, and then we have 2 in bats. And for our interest today, we are looking at the SARS-CoV-2, which falls under the beta coronavirus in mammals. Studies are ongoing to determine what are the intermediate hosts, but it has been shown that the bats are the natural reservoir for SARS-CoV. So in terms of the virus, when you are talking of detection and diagnosis, we are looking at the various proteins. So we have the outer layer proteins, famous one, the spike protein, and we also have the envelope protein, the nucleocapsid, which is forms the abundance viral protein. So these are the various proteins that we will use to detect either through the PCR method molecular or through the serological, which we are looking at today. So let's start with the basic terms. So in my talk, I'll be talking about the antigen or the antibody. So antigens are basically the foreign molecule structures that can trigger an immune response, for example, the SARS-CoV. And then the antibody is a protein, or also known as the immunoglobulin Ig, which is made by the body's immune system to recognize and target an antigen for destruction. So in terms of how to diagnose or detect the SARS-CoV, we use what's called the immunological testing of the SARS-CoV, where we detect either the intact virus, which is a direct method, or the indirect method where we look at the antibodies produced against the virus. So let's look at how we do the antigen detection test. So this is to detect the intact virus through the outer virus, as I mentioned in the previous slide, where we are looking at a couple of the viral antigens by using the specific antibodies. So here, for example, we get the nasopharyngeal sample collected, we treat using the transport media, and then we put into the selective antibodies. And then if there are presence of those viral proteins, it can be able to attach with the antibodies, and then you get a positive results. So this is more of a rapid test, which is quite easy, between 5 to 15 minutes. It's a well-established technique for other diseases. It has been used for HIV, for testing. It's a low cost, and also it can be produced in massive at the point of need. The limitation, though, for this is it has limited sensitivity. So we encounter a lot of false negative for the low viral load. And again, the responsibility is quite an issue between the pages, between the qualitative results and not also the viral load. So it's an ideal test for a massive COVID detection, but there is no solution available yet because these are quite new assays that are developed in the market. The second one is the serological test, which is an indirect method of testing for the presence of the SARS-CoV. So this one detects the antibodies produced by the infected person following the disease, either in blood or in serum. So again, here we use the rapid test, as I mentioned, which is a quite easy method, between 5 to 15 minutes. It's also a well-established method, easy to sample, extract, non-infective sample, low cost, and also it can be used at the point of care as required. Again, the sensitivity for these rapid tests are quite low. So we encounter false negative or the false positive. So this is ideal test for testing the presence of the antibodies or seroprevalence. But again, it has not met the required level of the sensitivity. there is no information yet about the duration and the quality of the immunity. I know Muli talk about production of the antibodies, the adaptive method, but again, we don't know how long these antibodies last in an individual. So immunity passport is not a good idea because we don't know how long these antibodies exist. So when we look at the kinetics of the antibody responses, we have three phases here. We have the incubation period, which is where we, between the days 0 and 5, individuals have been exposed to SARS-CoV, and then we are not able to detect the antibodies at that stage. At this stage, it is also a symptomatic stage. The individuals have not shown any signs of the disease. Between day 5 to 20, that is a phase where we have the disease. There is the onset of the SARS-CoV, as you can see in red. And then again, now from day 7 there and about, we tend to see different immunoglobulin classes appearing. The first one to appear is the IgM, which appears between day 5 and lasts for up to around day 20, and then followed by the production of the IgG and the IgA. However, the IgM wanes or decays quite fast. From day 20 and above, then we tend to have undetectable IgG, IgM, and IgA, while the IgG manifests for a long time. So because of that presence of the antibodies, there was a lot of hype. People are talking about we can be able to test the presence of the antibodies, and then it can open the economies, kids can go to school. But again, I tend to think that was too much hype because we put the cart before the horse, because we didn't know how much this antibody lasts. It can be 2 weeks, 3 weeks, a month, 2 months. So there's a lot of ongoing research to determine how long these antibodies can last in an individual. And again, there is the element of the protective. Can these antibodies confer protection in human, or they are just presence of the antibodies in an individual? So there are lots and lots of available serological tests for anti-SARS-CoV that were available from different companies. So these detection kits, they come in various forms. So there are a lot of variations about these test kits. One is because of the format. So some of the rapid tests, especially the ones that we have, like the pregnancy, the HIV kits, uses the lateral flow assays, where you can put the sample, either the blood, if you're looking for the antibodies, or the swabs, where you dilute in the transport media, and you can be able to detect the presence of the viral antigen. Again, there is another format, which is the enzyme assay. So this is a lab-based, whereby you have quantify the amount of the antibodies on the antigen present, or you can use also the chemiluminescence assays. The second component in terms of this assay variability is specimen type. What kind of sample do we use to detect the antibodies, or to detect the presence of antigens? So we use the serum, the plasma, finger pricks, or the whole blood, nasopharyngeal if you are looking for the antigen. Again, which class of antibodies are we likely to detect? Can we use the IgM, the IgA, IgG? Because again, these antibodies decay over time. They have different half-lives. And then lastly is for the SARS-CoV antigen. This antigen, that the rapid test that you are using, are they looking at the spike protein? Are they looking at the receptor binding protein, or they are looking at the abundant viral protein, which is the nucleocapsid? So those are the various considerations that we need to have in mind when you're choosing which antigen or antibody test to use for detection of the SARS-CoV. Mrna talked about how the presence of these antibodies confer immunity. But again, we need to understand there are two types of the antibodies. We have what's called the neutralizing antibodies, and then we have what's called the binding or the non-neutralizing antibodies. So protective immunity is multifaceted. So if you test the presence of the antibodies, either through the natural infection or through the vaccine, are these antibodies conferring protective immunity? So antibodies can be binding in terms of the non-neutralizing antibodies. So these are the antibodies that are produced generally in high levels as compared to the non-neutralizing antibodies. But these antibodies on themselves cannot prevent infection. It must involve other arms of the immune system in terms of talking of the innate or the T cells. These antibodies normally bind and flag the pathogen as an invader, and then it can happen what's called the natural killer cell cytotoxicity, or it can use what's called the opsonization of the phagocytosis. However, there are good markers of prior infection. So individual who has had prior or SARS-CoV, you can be able to detect this presence of the antibodies. And then the second component is the neutralizing antibodies, which everyone is quite interested in. These neutralizing antibodies, as Muli mentioned, bind the virus, leading to the loss of infectivity. So it prevents the virus from infecting other cells or entry into the host cells. Its function is independent. So once these antibodies are produced, they can be able on their own, without the help of other immune cells, such as the innate or the adaptive, to be able to attack or block the entry of these viruses into the host cells. However, the commercially available assays do not distinguish. So the assays that we have, the rapid test from different companies, all of them, they don't differentiate between the neutralizing antibodies and the non-neutralizing antibodies. So if you want to test for the neutralizing antibodies, it needs more complex assays. Again, these are the functional assays that ideally you need to use the live virus, or you use the pseudoviruses. So these are viruses that you need to have a BSL. So in terms of the point of care, and in terms of early detection or easy detection of these antibodies, you need these rapid tests for you to be able to analyze or detect these antibodies. So increasingly, some of the labs that are BSL-2 can be able to use the pseudotyped viruses as compared to the live viruses. So the pseudotyped viruses, again, as we talk of the spike protein, so these are a bit or a component of the virus, for example, the spike protein, which you attach into a vehicle, for example, this VSV, and then you can be able to use these viruses now in the normal BSL-2 labs that we have. The idea of these pseudotype viruses is a one replication cycle. So these viruses are not infectious. Once it infects the cells in the lab, then that is it. Yeah, that's why we can use it for in a lower lab such as BSL-2, unlike the live virus where you need a BSL for culture. So in terms of the SARS-CoV, what do we know about their neutralizing antibodies? The SARS-CoV, do they generate neutralizing antibodies? And can we compare to the other coronaviruses that we have had previously? So when we look at the common coronaviruses in some of the studies that have been done, they have shown that common coronaviruses, the IgG or the immunoglobulin G normally peaks within 2 weeks post-infection and decline over time, as you can see on these volunteers that were infected with one of the common coronaviruses. When they were rechallenged again at 1 year post-infection, 66% of the individuals were shedding more viruses, which none developed the calls. So there was element of the protective antibody levels throughout, which dropped at around 2 years. When you look at the SARS-CoV, so again, these are the SARS-CoV that we call them SARS-CoV-1, which arises around 2002, 2003. The antibodies were produced within 3 to 4 months post-infection, but again, they declined to a detectable level by 6 to 7 years. So when you look at the SARS-CoV comparing with the common cold, it looks like the immunity or the production of the antibodies from the SARS-CoV normally lasts longer than the general or the common coronaviruses. When you look at, again, the MERS-CoV, so these neutralizing antibodies remain stable at 3 years. So we hope, again, that the SARS-CoV-2 can generate neutralizing antibodies that can be able to sustain or have a longer half-life. So when you look at the protective immunity post-SARS-CoV, I think this was a question that came once you have been infected naturally with the SARS-CoV or natural infection, do you confer immunity from a secondary infection? So studies that had been done in the macaques showed that initial infection led to the binding and neutralizing antibodies to spike protein in all the animals, as you can see in the left graph. This macaques, they were rechallenged on day 35 post initial infection. And as you can see, at around day day 1 to day 2 of the rechallenge, there were low levels of the mRNA, which means there were low levels of the viruses, and there were no recoverable virus post day 2. So after day 2, as you can see here, none of the animals or the macaques showed any presence of the virus, and there were little to no clinical observed. So it means that post SARS-CoV initial infection, there is the element of natural immunity. Again, when they look at the neutralizing antibodies, they found that in 175 recovered patients, or patients who had the virus and they recover convalescent, there were a presence of the neutralizing antibodies. Again, it depends on our individuals. So the titers peaked between 10 to 15 days after symptom onset and were variable. So 5.7% of those individuals did not develop the neutralizing antibodies, while 30% developed neutralizing antibodies at low levels. So this is quite good. If individuals are infected with the SARS-CoV and they can be able to produce neutralizing antibodies within 10 to 15 days, then it can be able to offer the protection. So the unknowns are what are the neutralizing antibody titers that are clinically significant that can be able to protect individuals. And again, how long do these neutralizing antibodies persist in an individual? Is it 5 years? Is it 6 months? So the research are ongoing to determine how those neutralizing antibodies can last in an individual. So the immunoassays, or the antigen and the antibody test, how do they correlate with the neutralizing antibodies? So the commercially immunoassays are all qualitative, as I mentioned. Few studies published to date have shown that when you compare the levels of binding antibodies and comparing with the neutralization assays, can they be able to correlate? So published studies have suggested that there's a very high correlation, R2 values above 0.9, where when you have high levels of the IgG, ELISA, then you also have high neutralization components. So it means when an individual, even before you measure the neutralization assays I mentioned, it's quite challenging and complex and requires a lot of sophisticated equipments. Then you can use the commercial available assays, for example, the IgG, that there's a correlation. If there is high concentration or high levels of the IgG, then it means there's likelihood that that individual also might be able to neutralize the viruses. So how do we interpret the results from the antigen tests? So as I mentioned, there is the antibody and the antigen test. So first is the antigen test, where you are looking at a certain component of the virus itself. Either you're looking at the spike protein, or you're looking at the nucleocapsid or the envelope. How do you interpret those results? So when you get a negative result, because this is a 5 to 15 minutes, it means there is a likelihood that a person is not near peak infection. But again, at this point, this individual is still contagious. Insufficient viral protein to be detected by the antigen test. So at this point, when there is a negative result from the antigen, you need to confirm with a PCR. PCR is more sensitive, because what it means here is when an individual when the virus infects the cell, it has to undergo the replication cycle. At that point, there may be insufficient viral proteins to be detected by the rapid test. So at this point, you need to qualify the negative result with the PCR, which is a more sensitive method. And then when you have a positive results, this suggests active infection, and it means an individual is shedding the virus. What about the antibody test? So again, when you have a negative results, likely no prior infection or exposure to the virus, it means the individuals tested too soon following the infection or immunosuppressed patients may be negative. Again, I mentioned in the previous slides that the presence of the antibodies normally develop within 5 to 10 days of infection. So if you test quite early the presence of the antibodies, they may not be enough or insufficient proteins or antibodies that your test or your rapid test will not be able to pick. Or it's some individuals that have immunosuppressed. Again, they may be unable to produce, or the immune system are unable to produce the antibodies. And also there are some individuals, which is a small percentage of individuals, may not seroconvert at all. So again, someone is exposed to the SARS-CoV, they may be contagious, but they don't generate the antibodies. When you have a positive result, it suggests a recent or past infection. Again, this may be impacted by the local or the regional prevalence. So what these results do not tell us yet is when the patient was infected, whether they are shedding the virus, live or dead, whether the patients or individuals are protected against reinfection. So these assays will just tell us that there is the presence of these antibodies, but it does not tell us when this particular patient was infected and whether they're shedding these live or dead viruses. So we cannot use positive results from the antibody test to guide decisions regarding adherence to social distancing recommendation or the use of the personal protective. Those are things that we need to continue applying. So in a nutshell, what are the antigen tests? So the antigen tests are quite quicker, cheaper, but they are less accurate as compared to more sophisticated assays such as the PCR. These antigen tests will detect the presence of the SARS-CoV by binding to the viral surface proteins, which I mentioned are the antigens. Again, the interpretation. So when you get a positive antigen test, it means it's an active infection. So this test is used for diagnostics of an individual who has been infected with the SARS-CoV, and then the person is presumed to be contagious. And the negative does not rule out the infection, may be considered presumptive and confirmed with a molecular test. So in terms of the limitations, again, this antigen test there is no amplification step. So if you have very low viral load, then there is the element of running for the false negative. Best for symptomatic individuals are during infection, but when the viral load is quite high. So you need to use this test when you know this particular person is at peak viremia. False positive can occur, especially in population where the prevalence is quite low. So the benefits, again, as I mentioned, is quite rapid, 15 minutes, high specificity, but low sensitivity, may be useful for screening in high-risk congregate settings. The positive results informs the clinical decision or on infection. And then for the antibody test, again, this is used to assess the exposure, but not for diagnosis, unlike the antigen. So this one detects the body's immune response to SARS-CoV, either past infection, not active infection. The antibody formation takes around 2 to 3 weeks post-infection to develop these antibodies, especially the IgM and the IgG. So when you have a positive antibody test, it means previous infection with the SARS-CoV. It does not indicate the current infection, because if you are looking for the active viral protein, then you use the antigen test. The negative probably has not been infected with the SARS-CoV, or as I mentioned, it's too early in an infection to have detectable antibodies. Or again, if some of the people, small percentage, just do not seroconvert at all, or people with comorbidities such as immunosuppressive, then they don't generate these antibodies. So using these assays that I've mentioned, the antibody, we did a seroprevalence rate study among 17, across the 17 sub-counties in Nairobi, using one of the kits called the Untitled, where they are looking at the IgG and the IgM. And from this data, the household, so we sampled around almost 2,000 households, and we found that the overall seroprevalence in Nairobi County was 35%. Again, as you can see, depending on the sub-county, if it's densely populated, then we found very high seroprevalence. When you stratify the COVID seroprevalence by age group in Nairobi County, we found that the ages between from teenagers 10 to 49, there was high seroprevalence, which means these individuals were exposed to the virus, and in most cases, some of them were symptomatic and they didn't present the disease. So the key findings from this seroprevalence study was that the prevalence I mentioned was 35%, which means at least 1 in 3 persons in Nairobi had been infected by COVID in the 8 months since the first case. So this study was done in November. Persons aged between 20 to 49 years were 2.5 times more likely to have been infected by COVID compared to children less than 10 years. Again, children less than 10 years and persons above 60 years had the lowest infection levels. We found there were no significant difference in men and women in terms of infection. They were equally infected. The infection level was higher in the more densely populated sub-counties of Nairobi. And also half of the households in Nairobi had at least one person who had been infected. However, in most cases, the remaining people in the households had no evidence of past infection. On average, 3 in every 10,000 COVID-infected individuals died out of COVID. This finding indicates that, again, it was very low mortality as compared to the other developed countries where we had high mortality rate. And similar to other countries, the current surveillance system detects only around 2.5 of all the infections. So to bring you to what we are doing currently at KAVI, we have 2 studies on COVID or SARS-CoV. So one is to define the antiviral humoral immunity against the SARS-CoV in Kenya. So we have samples from individuals, the first wave and the second wave, March and in November. So we collected samples from individuals who seroconvert and recover the convalescent, and also individuals from that serosurvey who are asymptomatic. We want to look at 2 components. So we want to look at the humoral profiles of the symptomatic and asymptomatic. We want to see these individuals who they belong to a household that had someone who developed the infection, but they themselves, they didn't develop the infection. So we wanted to see the immune response. Can we be able to look at the antibody profile? And then in terms of how the phagocytose, in terms of the maturation, we also look at the cellular cytotoxicity of these binding antibodies involving the innate cells. The second component is the viruses. So we also get from these individuals, the serum or the plasma. We want to detect the presence of the antibodies that they have. Do they have the neutralizing antibodies or the non-neutralizing antibodies? Are these antibodies able to, are effective against the emerging global variants? We also want to see whether these serum or the antibodies from these individuals that have recovered can be able to protect against the Kenyan viral variants. So again, we collected the nasopharyngeal swabs that we can use to generate or isolate the viruses from these individuals. And then from the plasma, from the people who the convalescent plasma, then we can be able now to see whether these viruses can be able to either be blocked by the neutralizing antibodies or non-neutralizing antibodies from these individuals. Secondly is, that was the first study is looking at the natural infection. We also want to see the people who have been given the vaccine. Do they elicit the same immune response? So the second study is assessment of the COVID-19 vaccines immunogenicity and effectiveness in Kenya using the antibody-based surveillance. So the individuals who are given the vaccines currently rolled out in Kenya, AstraZeneca, then we'll be able to get the samples from the first dose, which acts as the baseline. We want to see whether these individuals were pre-exposed prior to infection. So this is nested on the national rollout. And then we get the samples there, the SST tube for the plasma and serum, and we also get blood, pyranized tubes, blood for PBMCs, to later on look at the adaptive immune system. So we'll collect at different time points to look at the kinetics of antibodies over time. So we want to see the individuals when they're given the first dose, 7 days, 14 days, 28 days. When they go for the second dose, we also want to get the samples from these individuals. And again, we want to see whether individuals who have previous or have been exposed to the SARS-CoV and they have recovered, or they were exposed and they didn't develop the infection, their immune responses, whether first dose is enough or the second dose is offering any other advantage or none. So we will, because for the logistic purposes, individuals in Kenya will be given the 2 doses. So for us, we want to prove the concept that only 1 dose can be enough for an individual, especially those who had been exposed to SARS-CoV and they have the antibodies, or do we also, individuals who were not exposed to SARS-CoV and didn't have the antibodies, can they be able to generate immune responses and see whether the first dose or the second dose can bring the difference? So for this assay, we'll determine the levels of the anti-SARS-CoV, IgA, IgG, IgM biolyzers that I've mentioned earlier. So the geek ups in our knowledge is which serological tests can identify the SARS-CoV neutralizing antibodies. As I mentioned, all the commercial available assays or tests do not distinguish between the neutralizing and the non-neutralizing. Is there cross-reactivity between neutralizing antibodies and other coronaviruses? Which SARS-CoV-2 antigens are optimal for detection of the neutralizing antibodies? And what is the correlation between the SARS-CoV specific antibodies and protective immunity status? How long does protective immunity last in recovered patients? are individuals susceptible to reinfection with SARS-CoV? I think this is a question that is still being researched by many individuals. And also, are these serological test antibody response the best indicator for protective immunity, or are there other immune cell-based mechanisms that we can be able to use? So I would like to stop there and answer the questions. Thank you. Back to you, Marianne.
Thank you very much, Dr. Langat, again for a very clear presentation, and we got you quite well, so thank you. There are several questions in the chat and also a few raised hands. I'll probably ask the people with the raised hands to maybe put the questions in the chat so we can go pretty fast. And I will allow a few questions because of time, and maybe we can start. Robert, you can see some of the questions. So we can start with some of the most burning questions, and let me just quickly go through them. In this era of vaccine-associated SARS-CoV-2 immune responses, is it possible to test and distinguish between the serological responses due to new viral infection and that of the vaccine antigen, Robert? Is it possible to distinguish these two types?
Yes, so as again, I mentioned, the sensitivity for these serological assays are quite low. But in most cases, when you're looking for the antigen, we have their specificity is quite high. So you can be able to distinguish between the SARS-CoV because they're looking at a certain viral proteins as compared to other new viral infections. So I guess the OIT is asking about other new viral infection that may not necessarily be SARS-CoV. So these are quite specific to the SARS-CoV because they are looking at a certain viral proteins.
Okay.
Marianne, maybe just to add on to what Langat is saying, this is Professor Nzala. You see, in an individual who has an infection, the antibodies are not just against the spike protein. The antibodies are actually against nearly all the proteins. So we can actually be able to tell an antibody response to a viral infection and an antibody response to a vaccine antigen, because the responses will actually be different. So it is actually very clear that we can unpack, the two. Even those individuals who had SARS-CoV and are given the vaccine, when you go in, you'll actually be able to see a multiplicity of antigens beyond just the antigens to the SARS-CoV, to the spike protein. Over.
Yeah, thanks for that.
So there's this also question, I think, thrown at everyone at COV, you out there. Why can't COV develop an antigen test for detecting SARS-CoV-2? Is it because of lack of funding or lack of infrastructure or lack of technology? Over to you.
I think, Prof, you can give a shot on that.
Okay, it's interesting. Or is Masika on the call? Is Masika on the call?
Yes, he is.
Okay, the development of these technologies is actually not difficult. What I can say, it is something that we are now beginning to put into process. We are developing a small biotech within COV to begin to think of making molecules and looking for vaccine targets, but also developing molecules to look for diagnostics. So just watch this space. In the next couple of years, you'll actually see a difference. And it's not just an issue of COVID alone. There are so many other pathogens that are of interest to us. We don't have a good test against dengue virus. We don't have a very good test against chikungunya. So there's quite a bit, but I'd like Masika to also say something about this. Masika? For me, this is the next generation that will do, the kind of things that you're asking. Masika?
Yeah, thanks, Prof. I think there isn't antigen tests, especially the rapid tests, are not so difficult to make. All we need is a system for expressing the antigens, which does not necessarily need to come from culture. It's just probably recombinant technology. So I think it's just because we've not been working on it. And usually for you to be able to launch a product quickly in a timely manner after such an outbreak, you need to have been working on it, on similar technologies, which you can then rapidly repurpose. But I think it's, as we discussed previously, something that is doable. Of course, funding is always a challenge, but that is usually at the last bullet on your listing challenges. First of all, we sort out the other problems, and then we can blame the lack of funding.
Okay, so yeah, that is, watch out for that space. So thank you for that, for those two answers. Maybe the last answer, several people are asking, this can go both to Daniel and Robert. How do you differentiate between non-neutralizing antibodies and neutralizing antibodies, and can they be produced all at the same time?
Maybe I can just say that the only way you can differentiate them is by doing functional assays, because neutralization is a function of antibodies. So you need to do functional assays, because if you if you are looking at binding antibodies, neutralizing antibodies also bind to the antigen of interest. So you'll be able to detect binding antibodies that are both neutralizing and non-neutralizing. But then to be able to identify the neutralizing antibodies, you have to do neutralization using either live virus or using some other tools like pseudoviruses that have been developed to be able to check for neutralization. I don't know whether Robert has something to add.
Yeah, the second component, someone asked, he asked about whether they can be produced at the same time. Yes, so in an individual, we know the non-binding antibodies are quite in higher levels. So we have a small segment of the small percentages of that individual can produce the neutralizing antibodies. But when we are doing the detection or the assay in the lab, we can't differentiate the presence of the neutralizing unless we do the functional assay, as Muli mentioned.
Okay, and a quick one here, Robert, thank you. Now that you're on the floor, maybe you can say one or two things about NO swabs. There is a question or two asking about how effective they are. There are some studies that show that actually screening stool samples is probably the way to go in the future. Any comments on that?
Yes, so what we haven't done, and most of the assays that have been developed have been looking at the antigen test, have been looking at the nasopharyngeal. I know the anal swabs or testing for these has just come quite recently. I think the first study or the test were done in China. So for us, we, as part of these seroprevalence, we also collect the stool samples, but we are yet to analyze them in the lab. So yeah, I don't know whether my colleagues can say something on that further.
Yeah, what we've done is that we've collected the individuals. We are doing follow-up from acute to recovery. We are also collecting stool samples. Actually, in a number of them, as well as urine samples. In a number of them, actually, stool samples and urine samples have actually come out positive. Not many, but this is not using the antigen test. This is using, the PCR test. But with PCR, really, you need to go, further and actually investigate whether the PCR positive results you're seeing, whether there's virus there that is actually infectious. So there have been positives both in urine and stool, but we'll need to do more studies to really appreciate whether the virus present is infectious. Over.
Yeah, Miriam,
there's one more question about.
Someone, Miriam, was asking about how can someone join the vaccine study, who to contact.
Yes.
Yeah, so you can contact the four of us here, myself, Muli, Miriam, Prof, or you can just walk to COV and then we will tell you more about the vaccine studies.
Thank you. And then there's one more question before we close the session about blood donation. So how long after receiving the vaccine or recovering from COVID, can one donate blood? Robert or Daniel, are there any studies showing this?
Not any I know of. Yeah, I would pass that to the rest of the panel.
Yeah, I'm also, I haven't seen that. But I tend to think once an individual is, recovered from the COVID, we are looking at, I can say 3 months plus, but I'm not quite sure about that. So I don't know.
Yeah, 3 months. Yeah, 3 months would be there because there has been the fear that one would actually transmit SARS-CoV through blood transfusion. But from the literature I've looked at, there's very little viremia. And if there is viremia, it is very, very early during the infection. So the guess here would be that after recovery from COVID, maybe you give yourself a bit of time, 3 months or so, before you actually donate any form of blood. But the fear is always that you may transmit the virus, but that fear is actually totally not there, because even in macaque studies, and macaques maybe are also not very good animal models, they have not seen much SARS-CoV-2 viremia. The spread that we see, although there is infection in the endothelial cells within the very many organs during the cytokine storm in stage 3, but at that time, maybe the cytokine storm is self-sustaining. Even when you look in the blood, you actually don't see much viremia. But after 3 months after recovery, I believe one should be able to donate blood without any major problem. Over.
Okay, thank you very much. There are still several questions in the chat, and as the panel, I think because of the time, and we've run out of time, we'll try and answer the questions as they come on the chat. But without further ado, I'd like to bring this session to a close. If there's any burning question or one question, this is the time to ask. As we thank every participant today who's joined in. We had over 1,000 participants, so thank you very much for sparing your time to learn with us. The very good questions that again will stimulate us to ask questions in the lab as we investigate and look at vaccines, as we look at the variants and try and get ahead of this pandemic. So again, we just want to highlight, this is the Lab Week that's been celebrated worldwide, celebrating all the medical lab scientists and pathologists and everyone who gets involved in the lab, which I think we all do, and all the work that is going on there. Thank you to the team at KOV and all the studies, as Robert has mentioned, and the team. If you want to participate in any of the studies as well, you're welcome to walk over. We also have bi-weekly Friday morning sessions at 7, where we discuss more of our research, where we hear more from our students, both at postgraduate level and postdoctoral sessions as well. So you're welcome to join us in those virtual sessions. So without further ado, I'll let Professor Nzala to give us his closing remarks. Thank you.
No, thank you, Marianne, and thank you, the KOV team, and thank you, the KNH team, for actually giving us the opportunity to talk about SARS-CoV. And it is still evolving, so we shall be glad to come back again, more so when we have data from some of our studies that are already ongoing on COVID. But something else that we are thinking of also is to have a webinar in the next 2 or 3 weeks to revisit HIV. Ladies, HIV has not gone anywhere. HIV is still around, HIV is still infecting. So as we talk about SARS-CoV, because it's the most current thing, we must also be current in terms of where we are with HIV. One of the major questions that is being asked in HIV is that what would it take to cure HIV? What would it take to cure HIV? And that is one of the discussions that we should be having in the next couple of, in the next couple of weeks. So I salute everybody. I thank everybody. I now give it back to the KNH team to give their closing remarks, and then we can move on to other things. Over to the KNH team.
Professor, and thank you everyone who has joined the session. I would want to invite you for the other sessions in the course of the week, and we look forward to all the discussions. Thank you all, and good night to everyone.
Good night.
Okay, bye-bye.
Bye-bye. Thank you, everybody.
Bye-bye.
All right, thanks everyone.